There are two main steps in protein synthesis: transcription and translation. This is a eukaryotic cell. The control center of the cell is known as the nucleus. Each cell in the body contains DNA. DNA is the genetic material required for replication and continuation of life. All DNA in eukaryotic cells are stored within the nucleus.
Transcription is the first step in protein synthesis. Transcription is the step in which mRNA is made by copying or transcribing the template strand. The template strand is the 5' to 3' strand, not the 3' to 5' strand. First, the enzyme helicase comes in, unwind the DNA strand, then proceeds to unzip the DNA strand by breaking the hydrogen bonds between the bases. Then, RNA polymers brings in nucleic bases that proceeds to the bases of the 5' to 3' DNA strand, then makes the sugar phosphate backbone. Finally, ligase comes in and closes any gaps in the backbones. This mRNA strand is called primary mRNA. Before it can leave the nucleus, it undergoes a lot of editing.
Within a gene, there are segments of DNA that are not part of the gene. These segments are called introns. They do not play a role in coding for protein. The other segments in the gene are called exons. Exons, unlike introns, do code for proteins. During the editing process, all introns are removed, so all we're left with is exons. The mRNA strand then goes through a bit of more processing, then is ready to leave the nucleus. The final edited mRNA strand is called mature mRNA, then leaves the nucleus through the nuclear pores. After leaving the nucleus, the mRNA strand goes to the rough endoplasmic reticulum. This is where translation happens.
There are three parts to translation: initiation, elongation, and termination. At the RER, a ribosome attaches to the mRNA. The ribosome is composed of two subunits, a large subunit made of protein and a smaller subunit made of rRNA. The rRNA portion of the ribosome contains a pocket. This pocket is where the mRNA stays. The pocket has two sites, a P site and an A site. Each site is limited to holding one codon at a time. A codon is a sequence of 3 nucleic bases that correspond to a code for an amino acid. Throughout the process of translation, mRNA stays in the ribosomal pocket. tRNA is the one that brings in the amino acids. On the tRNA, there is an anticodon, which is a complementary codon to that on the mRNA. The tRNA picks up an amino acid and brings it to the mRNA, where it attaches to the codon via its anticodon. To initiate, the first amino acid tRNA brings in is methionine. This is known as the initiation step.
Then the process of elongation begins. Another tRNA brings in another amino acid. The two amino acids bond, forming a dipeptide bond. Then the tRNA in P site leaves and the ribosome shifts over 1, so that the tRNA originally in the A site is now in the P site. So another tRNA comes into the A site and bringing another amino acid. The amino acid forms a polypeptide bond.
This continues until the stop codon is reached. This is called termination. It's important to note that there is no tRNA that corresponds to the stop codon, and instead there is a protein that comes in called a release factor. This releases the polypeptide chain from the last tRNA and hydrolyzes the ribosome. After the polypeptide is released, it folds into its secondary and tertiary structures, and then moves to the Golgi body to be modified and packaged.